Hello everyone, welcome to my channel Anupma Biology Classes. In this video, we will discuss about the kingdom Protista. Before this, we discussed about kingdom Monera. This video is taken from CBSE class 11th, part 5th of chapter 2nd. Now, let's start the topic. So, first thing is, what is kingdom Protista and which type of organism are placed in this kingdom? So, kingdom Protista, in Greek, Protista means first of all a primary organism. So, these includes all unicellular eukaryotic microorganism. So, before this, in the kingdom Monera, we discussed about the prokaryotic organism. And prokaryotic means they have absence of true nucleus. Now, in this organism, from the Protista, all organisms are eukaryotic, that means true nucleus present, but they all are unicellular. So, all organisms in this groups are unicellular eukaryotic microorganisms except some unicellular algae like Chlamydomonas, chlorella, etc., some fungi, yeast, and animals which are placed in their respective kingdoms along with their closely related multicellular organism. Usually, leaves Protista lives as solitary cells, but a few forms may develop colonies of similar cells without much cellular differentiation. They are predominantly water dwelling and live wherever there is water. Phylogenetically, the kingdom Protista acts as a connecting link between the prokaryotic kingdom Monera on one hand and the complex multicellular kingdoms of fungi, plants, and animals on the other hand. The protists are regarded as ancestors of all multicellular eukaryotic organisms. Now, what are the general characters of the kingdom Protista? So, first thing is their habitats, and after this, we discussed about the protist structure. So, what are their habitats? Protists are mostly aquatic organisms and live wherever there is water, in the sea, in fresh water, and in moist soil. Some live in the bodies of animals as parasites. Most of the protists inhabit the surface of water bodies in the form of planktons. Now, if we talk about the protist structure, protists are distinctly microscopic unicellular organisms. However, some individuals form colonies of similar but largely independent cells. The unicellular protists have different shape and size. The cell's structure is typically eukaryotic. So here you can see this is a eukaryotic structure. Now, in case of their structure, if we talk about the protist structure, the protoplasm is surrounded by a distinct plasma membrane or plasma lemma. So protoplasm is present inside the inside the cell, and the membrane is the outer structure which bound the protoplasm inside. Here no cell wall present. In addition, some protists have an outer covering of pellicle, cuticle, cell or cellulose wall. The photosynthetic forms have a distinct cell wall made up of cellulose. Internally, the cells have distinct membrane-bound organelles like nucleus with chromosomes, mitochondria, Golgi bodies, endoplasmic reticulum. Endoplasmic reticulums are of two types, smooth endoplasmic reticulum and rough endoplasmic reticulum. Ribosomes, which are of 80S types in this, because you know that in case of prokaryote, 70S type of ribosomes present, which are found in monerans. Now, in this kingdom, all are eukaryotic, so 80S type of ribosomes present, and chloroplasts, etc. So, the photosynthetic protist possess chloroplast with the internal thylakoids. Some forms contains centrioles in their cytoplasm. The nucleus consists of chromatin, nucleolus, and nucleoplasm which is surrounded by porous nuclear envelope. The genetic material is linear DNA. The chromosomes here possesses centromeres for attaching to spindle fibers during mitosis. Cytoplasm remains in a continuous state of motion, that is, so cytoplasmic streaming. Some predatory protozoans have special organelles for the intake of food and elimination of waste. Some motile protists many have flagella or cilia for the locomotion. So, in case of flagella, they have typical 9+2 pattern of fibril, that is an external cylindrical of 9 doublet fibrils in the periphery and a pair of single fibril in the center. The fibrils are finally made up of a protein which is known as tubulin protein. Now, locomotion in protist. Locomotion is the movement of an organism or its part from one place to another. Motile protists move from one place to another place with the help of Pseudopodia, Flagella or Cilia. Sometimes a few of them by wriggling or and mucilage propulsion mechanism. So, locomotion by Pseudopodia, locomotion by Flagella, locomotion by Cilia, wriggling locomotion and locomotion by mucilage propulsion. So these are the type of locomotion which are found in case of protist. Now we will see each and every part in detail. So the first one is locomotion by Pseudopodia. Some protists like amoeba slowly creep over the fast substratum with the help of blunt finger-shaped protoplasmic extension called pseudopodia or false feet. So in this picture you can see the cursor indicate this is the pseudopodia or this is also known as false feet. They give irregular shape to the organism. The rate of movement is 0.2 to 3 micrometer per second. This type of locomotion occurs in several protozoans, that is sarcodinas, amoeba, etc. and cyme molds also. So pseudopodia are of 4 types. First one is here. What are the types of pseudopodia? Lobopodia, philopodia, reticulopodia and exopodia. So see here, this is the lobopodium. Here the name indicate. So lobopodium is a lobe-like with broad and blunt ends. It's found in case of amoeba. Next is philopodia. Philopodia is a fine thread-like, often with rounded ends, and it is found in Euglypha. Next is here the example reticulopodia. So reticulopodia is a long and branched forming network and is found in Globigerina. And the last one that is known as exopodia, which is found in actinophytes, and this is long and stiff with a hard axial filament. So there are 4 types of pseudopodia, and these are lobopodia, philopodia, exopodia and reticulopodia. Now locomotion by flagella. Flagella are motile, hair-like appendages. Okay, with projects from the free surface of the cells. So the external long, see here, external long and whip-like part of the flagellum is called shaft. It varies in the length from 2 micron to 3 millimeter. The diameter remains almost uniform, about 0.2 micron. Internal structure of the flagellum consists of 9+2 fibrillar arrangement. Most flagella exhibit undulating movement, which propels the organism in the direction opposite to that of the beat. Some protists have only one flagellum, like in case of Euglena. In this picture you can see, while others have more than one, up to 4. The rate of movement is 15 to 300 micrometer per second. Locomotion flagella occurs in case of dinoflagellates like Gymnopodium. Euglenoids, Euglena, and Jufladlets like Lesmania. So there are three where you can see the locomotion by Flagella like dinoflagellates, Euglenoids, and Jufladlets. After this, locomotion by cilia. Cilia are short, fine hair-like structures present all over the body surface in large numbers. Here you can see this is the cilia-like structure all over the body. As compared to flagella, they are much smaller but produced in large numbers. So cilia are produced all over the body. Here you can see like hair-like appendages. But the flagella is only one and present on an end mostly. They perform oar-like movement in a coordinate manner like a group of persons rowing a boat and produce a progressive wave by beating in succession. The cilia-bearing protists are called ciliates. They are the fastest among all predatory protists and move at the rate of about 2 mm per second. And the example is paramecium. So paramecium is an example of locomotion by cilia. Now, what are the differences between the flagella and the cilia? Cilia are short, hair-like appendages extending from the surface of a living cell, while the flagella are long, thread-like appendages on the surface of the living cell. Nexine arm present in case of cilia, while the nexine arm absent in flagella. Cilia are short, while the flagella are longer than cilia, can vary. Cilia have rotational like a motor very fast moving, while the flagella wave like undulating sinusoidal slow movement compared to cilia. Cilia are present in many, that means in hundreds per cell, while the flagella are few, less than 10 per cell. Cilia present in eukaryotic cells, while the flagella present in eukaryotic and prokaryotic cells, both, because you know that in case of bacteria, we discussed about the flagella and the type of flagella which are present in case of bacteria. So, flagella are found in eukaryote and prokaryotes both, while the cilia are present in only eukaryotic cells. Now, wriggling movement. So, what is wriggling movement? It is a kind of slow worm-like movement of non-flagellated protists. That means they have absence of flagella. And they perform with the help of wave of contraction and expansion of body. An example here you can see that is the sporozoans, non-flagellated euglenoids, etc. Now, locomotion by mucilage propulsion. Some protists like diatoms exhibit creeping or rolling type of movement by secreting soft mucilage in the direction opposite to the direction of the movement, and this is known as locomotion by mucilage propulsion. So these are all about the locomotion. Now nutrition in protist. So kingdom Protista includes diverse type of unicellular organisms which either derive their nourishment from their surrounding or prepare their own food. So the organism perform the following modes of nutrition like photosynthetic or holophytic, holozoic, saprobic, parasitic and mixotrophic. So see here each and every part. First one, that is the photosynthetic or holophytic. Many protists possess photosynthetic pigments like chlorophylls and perform photosynthesis. They prepare their own organic food from carbon dioxide and water with the help of photosynthetic pigments, making use of sunlight as an external energy source. The photosynthetic protists are dinoflagellates like gymnodinium, diatoms like pinularia, and euglenoids like euglena. After this, holozoic mode of nutrition. In holozoic, holos means whole and jun means animal. So in case of holozoic mode of nutrition, the individual captures, ingests, digests and assimilates other animals and plants. The holozoic protists are mainly protozoans. Example, amoeba. In this picture you can see, and paramecia. After this, saprobic mode of nutrition. In this mode of nutrition, the protists live upon decaying organic matter. They release some extracellular enzymes into the surroundings where the enzymes break down the complex organic molecules into the simple forms. The simple soluble substances are absorbed through body surface of the organism by diffusion. The saprobic mode of nutrition occurs in case of slime molds. After this, parasitic mode of nutrition. In this mode of nutrition, the protists obtain their nourishment from other living organisms. And the examples like monocystis, plasmodium, giardia, entamoeba, etc. After this, the mixotrophic mode of nutrition. In this mode of nutrition, the protists obtain their nourishment by more than one method at the same or at different time due to change in external conditions. For example, mode of nutrition, which is mixotrophic, is found in Euglena, which takes its nutrition by holozoic, holophytic and saprobic method. Facultative heterotrophs or facultative parasites are also sometimes referred to as the mixotrophs. So they have both type of nutrition. So they are known as mixotrophic. After this, now see here the reproduction in case of protist. So reproduction occurs both by asexual and sexual method. In case of asexual, there are some other options like the binary fission, multiple fission, plasmotomy, budding, spore formation, etc. And the next one is sexual reproduction. So we will discuss about it in the next slide. See here, in case of asexual reproduction, it is the most common method of the reproduction in protists, in which the genetic constitutions of the young ones remains the same as that of the parent. Under favorable environmental conditions, they reproduce asexually several times a day, resulting in population explosions. The major types of asexual reproductions are, first one, that is the binary fission. In this picture you can see the parent cell divides into two approximately equal daughter cells, either transversely like in paramecium. Here you can see the transverse binary fission, longitudinally like in case of Euglena, or axially like in amoeba by mitosis. So longitudinal binary fission occur here in Euglena, transverse binary fission occur in paramecium, and irregular binary fission or axially occur in case of amoeba. After this, multiple fission. Division of parent cell into a number of daughter cells is called multiple fission, and it is occur in case of plasmodium. Next is plasmotomy, fission of multinucleate protist into two or more multinucleate offsprings by the division of cytoplasm without clear division is called plasmotomy, and it occurs in case of opelina. In this picture you can see. So plasmotomy is a special type of fission in which a multinucleate protozoon divides into two or more multinucleate daughter individuals. This is the structure of. Now, this is the structure of opelina. Now see here, budding. In this type of asexual reproduction, a small bud is formed from the parent body, which separates and develops into the new individuals like Parasinata and Cilia. Now, see here, this is a spores formation. Sessile or stucked sporogonia containing spores are formed in slime molds. They liberate the spores which can withstand a prolonged period of desiccation. On germination, each spores give rise to a new individual. And this you can see in case of the slime molds. Here is spore formation and spore dispersal. Now, so this is all about the binary fission, multiple fission, plasmatomy, budding, spore formation in case of the asexual reproduction. Now, sexual reproduction. Sexual reproduction is believed to have originated in the primitive protist. It involves meiosis and syngamy. Meiosis means reduction division and syngamy means fertilization. So during meiosis, the chromosome's number of parent cell is reduced to half, that is 2n. Diploid number of chromosomes in the parent cell becomes haploid, 1n number in gamete, which is known as special sex cells. So here you can see that this is the gamete which have one nucleus, and before this is haploid in condition, and before this adult is in 2n condition, this means diploid. The two gametes having 1n number of chromosomes fuse to form a diploid 2n zygote. Here you can see this is the 2n zygote. So the first, here after the meiosis, here you can see that a young individual after growth converted into the adult, which is 1n condition. Then mitosis occur and two fuse to form gametes, and after fertilization between the two forms a zygote which is in the 2n condition. Then after that meiosis occur and the young individuals form. So fusion of gametes is called syngamy. And the syngamy occurs through isogamy, anisogamy and oogamy. So isogamy, what is isogamy? Isogamy means the two fusing gametes are structurally and functionally similar, like monocystis. So different genotype but both are in the same structure and function. Anisogamy. So what is anisogamy? The two fusing gametes are similar but differ only in their size and or motility, like seratium. And oogamy, that is large non-motile gametes are fertilized by smaller one gametes like the plasmodium. This is known as the oogamy, and a different motility. Heterogamy or anisogamy means different size, and isogamy means different genotype. So what are the differences in anisogamy, isogamy and oogamy? Anisogamy is the fusion of gametes in the dissimilar size, while the isogamy is the fusion of gametes in similar size, and oogamy is the fusion of large immotile female gametes with a small motile male gametes. In case of anisogamy, male and female gametes are present. In case of isogamy, male and female gametes are not distinguishable. And in case of oogamy, male and female gametes are present. In case of anisogamy, both male and female gametes are either motile or immotile. In case of isogamy, both male and female gametes are either motile or immotile. And in case of oogamy, egg cell is immotile and the sperm cell is motile. In case of anisogamy, second stage of evolution in the sexual process. In case of isogamy, the first stage of evolution in the sexual process. And in the oogamy, third stage of evolution in the sexual process. Anisogamy occurs in some fungi, higher in vertebrates and all vertebrates. Isogamy found in Chlamydomonas, which is a unicellular alga, and Monocystis, which is a protozoan. Oogamy found in higher groups of algae like volvox and oidogonium, plants like bryophytes, ferns and gymnosperms, protists and animals. So these are the differences between the anisogamy, isogamy and oogamy. Now, major groups of protists. So what are the major groups of protists? Unicellular protists have been broadly divided into three major groups. First one is the photosynthetic protists, which is known as the Protistum algae. Second is slime molds, which is known as the consumer decomposer protist. And the third is protozoan protist. So in the next video, we will see each and every groups of protists in detail. So this is the end of video. If you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any queries, any questions or any suggestions, you can ask in the comment section below. Thank you for the watching.